Welcome back. In this video, I will go over how to use the inverted light microscope to visualize the structures of materials. This is the inverted microscope setup. From top to bottom, we have the light source here to illuminate the sample on the stage. So this is the stage, and we usually put samples here. And here we have eyepiece, and under here we have some objectives. So we have objective lens here. This equipment is the camera, and this knob is the focus knob for us to tune the focus. This knob tunes the amount of light that goes through the sample. If you look at the microscope on the right, everything is similar, where we have the same focus on the right. It doesn't matter which focus you tune, they are all the same. We have the coarse focus, which is the larger one, and the fine focus, which is the smaller one. Usually we adjust the coarse focus first, then the fine focus. And the stage is connected to a joystick, and changing the knobs here will allow us to change the position of the stage. To turn the microscope on and off, you need to use the on and off button that is hidden behind this stage. Here is the on and off button. To turn on or off the microscope, you'll need to use the button hidden behind the microscope. You'll just need to switch the on-off switch on to turn it on. To start the computer software, click on the Micromanager software button. Then click OK before you click Live. Now we can start do some basic operations on the microscope, such as adjusting the light, focus, and stage position. The green light indicates that the light is currently off. To turn on the light, press the button so the green light is turned off. To adjust the amount of light that goes into the sample, you can use the slider. To increase the amount of light that goes in, slide up. To decrease the amount of light, slide down. You can verify this by looking at the sample stage to see whether or not the light has been turned on and increased and decreased. You can also see it on the computer to see how the light shifts, to increase the light and decrease the light. We can also tune the focus and stage position. For the focus, we'll first adjust the coarse focus to find the rough plane and then adjust the fine focus. For stage position, we can move the joystick. The upper one moves the stage in and out, and the bottom one moves it left and right. Now we're all set to calibrate the microscope. Put the calibration standard on the stage. Then we'll need to adjust the focus, the lighting, and the stage position to find the standard. First, let's adjust the lighting. Shift the lighting up and down to find the optimum position for the lighting. Sometimes it will overexpose, sometimes it will underexpose, but you want to find the perfect medium where it appears roughly gray when it's out of focus. Keep adjusting. Now we're in a gray zone. So now we want to adjust our focus. First use coarse focus, then use fine focus to finally pinpoint on the plane that we're wanting. And then move the stage position to position at the calibration standard. Then adjust the fine focus so that we can see the lines perfectly. Now after we find the line, we can then measure the distance between the lines. I'll go to image J and select the tool for straight line. So first select the straight line tool to select the line between them, and go to Analyze, Set Scale. And each of this line has a distance of 10 micrometers. So those pixels corresponds to 10 micrometers. Now we can correspond the actual distance with the computer distance. And press OK. We also want to look at the scale bar. So go to Tools, Scale Bar, and then adjust the settings. Maybe we want 100 micrometers of scale bar. Position the scale bar and then do an overlay. Press OK. This is how we perform calibration as well as making images. We can go to File and save the image as JPEG or PNG as needed. Note that usually at a 10x objective and 1x eyepiece, we usually have a calibration around 2.9 pixels per micrometer. After taking the images, we can turn the microscope off. First, we want to turn the light off by pressing the button so that the green light is on. We can then turn off the microscope by switching off the button. Now let's look at how we can use the microscope in the surface and colloid lab. We can use it to look at porous structures of filter paper, and in this case I'll demonstrate looking at the structure of CHEM wipes. Here we adjust the coarse focus first to look at roughly the plane that the CHEM wipe is at. Then we'll use the fine focus to adjust to see the CHEM wipe's actual structure. We can also use the light microscope to look at the structure of emulsions, whether the big droplets or the smaller ones, and the fractal structures between them. And we can use image processing to look at the diameters of each large and small droplet sizes. In this video, I walk through the practical aspects of using an inverted light microscope to look at the structure of porous medias and emulsions.